The big one, but it'll
Can everyone hear? Who's introducing? It's here. It's here. Introduce yourself. Okay. So thanks everyone for humor. That's the right word. And I apologize for the multiple feedback. Clearly, I'm not cut out for events. So I felt it's about time I told everyone what I'm doing in my other life, which is not happening here. These are experiments that I've been involved in since I started my sabbatical in 2019. It was an aborted sabbatical because in the middle of COVID happened and I had to go home. But the experiment continued, and this part that I'm going to talk to you about is already published. It's coming out soon, so I'm going to tell you about that part of the story. The part that we're continuing, if we have time, we can discuss it. So the work I'm going to tell you about, I like to actually acknowledge people first here because I will forget at the end. This was done by a grad student who now is graduated and 4 postdocs who were involved in various aspects of this work. These two, Junichi and Leila, were directly working with me, so I was involved in all their experiments and what they did and all that stuff. And a lot of that is the behavior and fibrophotometry stuff that I'm going to point out later on. All of the experiments were done at the Einstein Med School, which is in the Bronx, and we're still continuing this work. Junichi has moved on to Yale. Leila is still working on that stuff, and there's another person, another postdoc who's going to soon start on this particular project. So the gist of this project, this kind of goes back, for those of you who were at my presentation this summer, to how animals produce behavior. So for all behaviors to actually happen, the animal has to kind of, not the animal, but the nervous system of the animal has to ensure that the movements that are going to happen are optimal and smooth, and that they're worth doing. So it has to kind of evaluate what things to do and what things not to do. And a big part of that involves a system that kind of, in a funny way, is now called the reward system of the brain, which basically evaluates things that are worth doing and allows that to go through. And in part, every movement that any animal, including us, does has a lot of planning and prediction to do before even the movement happens and while the movement is happening. And for that reason, the motor system is a huge part of the central nervous system. Now, the motor system in mammals, which is what I'm going to mainly focus on, involves, when you do the textbook version of this, 2 main components, which is the upper motor system, which in mammals we say is maybe the motor cortex and the premotor cortex, but it includes other components such as the tecton areas, control a lot of the movement. And then the lower motor system, which is all in the spinal cord, which commands the neurons that go to the muscles, or the motor neurons that do the actual movement. But even that part of doing movement requires enormous amount of computation. However, when you look at the parts of the brain that are involved in movement in terms of motor systems, there's two other components of the brain that come in that are involved in planning and executing movements. These are the basal ganglia and the cerebellum. These two components do somewhat different things. At least what I'm going to tell you now is the textbook version of this. Whereas the basal ganglia basically decide which actions should be done at this time and which actions should be stopped at this time. So you say they gate the movement, or they're the go, no-go parts of the central nervous system. In contrast, the cerebellum ensures that actions that are actually going to happen kind of in the best way. They happen smoothly and optimally. So if you screw up the cerebellum, you can still do stuff. The animal can actually do stuff. It just does it very, very, very poorly. Right? So if you think about it from a context of, an animal living in its environment, survival really depends on doing things fast and smoothly and often. If you want to, take your time, you get eaten, or you won't get food or mates, and you will not survive. Now, I also want to mention that the basal ganglia, which is they sit deep in the brain, and the cerebellum, they're both older parts of the brain than what we are considering here in mammals. Especially the cerebellar structures and cerebellar-like structures are present pretty much in all vertebrates, but they do a lot of different functions too. In particular, the cerebellum is involved, for example, in sensory processing. In fish, the cerebellar-like structures are mainly considered as sensory processing things, where they basically take. The main idea that has been shown in fish very elegantly is that they take the sensory component of what the animal is doing itself and subtract it off from the movement so that other sensory inputs that are coming in stand out. And the main point I want to make is that both of these structures are involved in sensory processing, and at least at the mammal stage and human stage, they are known now to be involved in higher cognitive processes. Okay. Now, there's a couple more things. I've already said this, so the only thing that I didn't mention, maybe I should mention, is that a big part of what the cerebellum does in us, for example, but in a lot of animals, it's just maintaining balance. The vestibular system is the sensory component of that, telling you where you are. And also the proprioceptive system, getting information from all your muscles and joints, etc. All of these inputs come to the cerebellum, and the cerebellum basically continuously commands how these muscles are acting. Now, there's one more thing that both of these components do that sits at the deeper level, and that is that both of them are involved in learning. So the basal ganglia basically are involved in what we call reward association learning. Reward is a loaded word. It's kind of one of these psychology words that entered into biology, but the term that is nowadays used, in fact, is also not from biology. It comes from computer science, which we call it reinforcement learning. And that is that you do something and you learn from that outcome, you decide to do it better and better, or not to do it, etc. So for example, learning to do any type of sports, I'm sorry, not sports, learning to do anything that gives you a reward, like teaching my dog to fetch, that requires repetition, and the reinforcement happens for the next file. So whereas the cerebellum, in fact, is involved in a different type of learning, and that is what we call the error correction or feed forward or supervised learning, in that the action that is being done, if there's a mistake in that action, it's corrected at that time. And yet, that type of learning, that error correction also is learned so that it becomes habitual. So as such, any type of sports learning is not related to the basal ganglia, but related to what the cerebellum does. Okay, doing things smoothly and optimally is done by the cerebellum. Now, there's two more things I want to say in the background, and then I'll go to the video. One is that for the basal ganglia to do their work, they require dopamine. Dopamine is a neuromodulatory substance, right? So it's just a neuromodulator that is released. In fact, pretty much all animals that we know have dopamine. In the mammalian brain, it's released mainly by the centers in the midbrain, and two of which are known as the ventral tegmental area, which sits in the tegmental. Right next to it is this structure known as the substantia nigra, where the core of it, which is called the compact part, or the pars compacta, are mainly dopaminergic neurons. They're 90%, 95% neurons that release dopamine, right? And they release dopamine, and I'll tell you where in a second. The ventral tegmental area does things in parallel, and I'll mention it briefly in a bit. But the dopamine release from the substantia nigra, that gets released in a structure known as the striatum. The striatum is a big nucleus in the middle of the brain. In fact, in humans, it's really two different nuclei called the caudate and the putamen. In mice and rats and rodents that we study, they're kind of together, and we call it the dorsal striatum. That is the main structure that all the inputs to the basal ganglia come. So that's called the input nucleus of the basal ganglia. Without dopamine in there, nothing will work. Okay, so dopamine is continuously coming in and modulating what is there. The main neurons in the striatum are these medium spiny neurons, right? These neurons project out of the striatum into different structures of the basal ganglia. For those of you who care, they're GABAergic neurons, so their actions are mainly inhibitory, and they get heavily modulated by dopamine. One of the things that happens that is a disease problem, that is a pretty serious one, is that in Parkinson's disease, this dopaminergic input is lost. Okay, it's lost gradually in that the neurons die. First, their processes, their projections in the striatum starts deteriorating, and eventually the neurons themselves are lost. So Parkinson's disease is really, its hallmark is the loss of dopamine neurons. And Parkinson's is really devastating in that it is the second most prevalent mental disease, not mental, disease or disorder of the nervous system that is known after Alzheimer's disease, okay? So the cause of Parkinson's is not known. So the dopamine neuron loss is not really the cause. That is one step that seems to be the hallmark. And the main therapy that is known with Parkinson's disease is to try to replace the dopamine. So I'll come back to that at the very end and talk about that a little bit. Now, what happens in Parkinson's? In Parkinson's, people lose their ability to initiate movement properly. They can't do it rapidly. Even when they manage to move, they can't move fast. They're much, much slower in their movements than the normal person is. There's another important thing that I didn't mention to you is that these two structures that I told you about, the basal ganglia and the cerebellum, even though they're considered in textbooks as part of the motor system, neither of them communicates with the spinal cord. They don't actually communicate with the neurons that are going out. They both instead get their inputs mainly from the cortex in mammals and send their outputs back to the cortex, right? So they are involved in these cortical loops. So this is shown in the cerebellum, where the input comes from all parts of the cortex. Input comes to the cerebellum, and from the cerebellum, it goes back to all parts of the cortex. So the cerebellum is kind of this unique structure. It's almost like a parallel brain that you have that gets every sensory input that you can name. and every input from every part of the cortex that you can name, and provides output to all of that, and does something. The basal ganglia similarly get their input in the striatum mainly from the cortex, although I told you they get modulated by the substantia nigra. The input comes from the cortex. And eventually, when the output goes out to the thalamus, back to the cortex. So they have this cortical loop that they're interacting, communicating with the cortex. Our hypothesis when we started this project, I'll give a little bit more about this, was that in addition to these cortical loops, the cerebellum is directly communicating with the dopaminergic centers in the brain. Now, I'm specifically interested in the substantia nigra as the dopaminergic center in the brain, but there was some precedence in this, in that the lab of my colleague, Kamran Khodaha, who did this, had already published that the cerebellum has direct communications to the other dopaminergic center in the brain, the ventral tegmental area, and that this input that goes from the deep cerebellar nuclei to the ventral tegmental area is involved, at least in a set of behaviors. I'll show you that in a second. Before I forget, the deep cerebellar nuclei are the output nuclei of the cerebellum. So almost all of 99% of the output of the cerebellum comes out from these nuclei that sit within the cerebellum, right, and project, out. Now, what they showed is that activating this pathway is rewarding, and they did this in a simple test. So you put the animal basically in an arena. You have these optic fibers in their brain, and you basically shine blue light, blue laser light, to activate these neurons that you've labeled with this genetically encoded construct known as channel rhodopsin. Channel rhodopsin, when you shine blue light on it, it depolarizes the neurons and it activates it, produces action potential. So the neurons are labeled, the deep cerebellar nuclei, with channel rhodopsin, and then the optic fiber is put on the VTA. So only the ends of these neurons, the axon terminals that are going to the VTA, are activated by channel rhodopsin. You shine light, and what you find is that the animal likes to spend its time mostly in the quadrant of this arena. The quadrant is only defined for us, not for the animal. But when the animal moves in there, it gets activated, right? And the animal likes to spend its time there. And there's additional experiments, but I'm highlighting two of these. This is what's called a 3-chamber test, and in which the animal is put in the middle. On one side, there is another conspecific. It's another mouse basically. And what the animal likes to do is like to go and spend its time with the other mouse, compared with an empty cage on this side. But when the animal goes in, you inhibit these neurons. So in this case, instead of exciting them, you express a different construct that inhibits these neurons. What you find is that the animal spends equal time on both sides. So it seems like the activity of these neurons is sufficient for the animal to want to spend time in a location, and is necessary for it to do this surveillance. That's basically what they had found. They hadn't gone too much deeper than that in that paper.
In the left here, is it trained to spend time in that quadrant?
No,
Is that the natural?
no, it just runs around. This is its normal, and then in 10-minute intervals, you just, whenever it goes in that quadrant, the camera catches it and just stimulates the axon terminals. So it likes to then spend its time there. And then when you can shut that off, and then it goes back to this behavior. This is the graphical abstract that I cut out. This is not the actual data. Okay.
The fact that the optogenic construct will actually serve to train them. means
So that is the stimulus is the training.
The stimulus is the reward, right? But this is not being done enough for the animal to actually learn it, because that has to be done, especially with mice that are dumb. The rats are kind of smart animals, but one of the things I realized is it is so painful to do behavior with mice because they're just dumb. It's not a very smart animal. So it's like this switch from rats to mice has been a huge detriment to studying animal behavior.
Did you come and present this?
Yeah, he came here and presented this. Yeah. In fact, Gal invited him and he came and presented this at our
Old club. It was, I think, in 2018. Yeah, that was in the first year.
yeah. Okay, so there's, I want to say that there's also historical evidence from anatomy papers that such connections may exist. And in fact, in 1959, this guy Carpenter looked at monkeys and what he had done is that he had ablated the deep cerebellar nuclei, and then he sliced the brain and looked around and saw degenerating fibers within the SNC. And that was replicated later in cats. And then there were a couple of studies from this guy, Watabe Uchida's lab, that basically did a tour de force with modern techniques of doing anatomy, and they mapped out all parts of the brain that project to the dopaminergic centers. So they just basically went in, labeled these, did light sheet microscopy on, different slices of the brain and just mapped everything. And they, if you look in their map, somewhere they also mark that the cerebellum is also projecting there, okay? It's also, oh yeah, I actually took this out to show you kind of what they did with the light sheet microscopy, which is pretty damn cool. They find the neurons, it's all automated. It's labeling and finding and marking and all of that. So this group did MRI, what do they call it? The something tensor imaging. Diffusion tensor imaging, thank you, looking at flow, basically water molecules, right? And they showed that this pathway actually exists and is active in humans. So that there's some evidence for that being functional in humans. Oh, there's also a couple of old papers. I seem to drop my end parentheses a lot, right? That showed that electrical stimulation of the cerebellum, if you stimulate the hell out of it and look at the striatum, the levels of dopamine go up for several hours. But the mechanism of that wasn't clear how or why. So from here on is data, and there's a lot of data, so I'm going to walk you through it and show you what we did. So in this case, we did the thing that I just told you. We, in the deep cerebellar nuclei, we basically injected these adeno-associated viruses that express channel rhodopsin, and there's a fluorescent protein attached to it. So you can look where you put this stuff. So you infect here, right? And then you take it out, you let the animals be for a few weeks, right? So this expresses itself in those neurons. And then you embed, in this case, what this is called an optrode. So this is both an electrode and an optic fiber. You embed that over the SNC, and you can actually both stimulate with a laser light. So these are 1 millisecond pulses of blue laser to activate the channel rhodopsin in the terminals of the cerebellar projections in the SNC, and also record what's happening. So this is what you see. So this is a single 1 millisecond blue light. And what you see is that when you turn it on, you get the output. This is a zoom in of that part, right? Which shows action potentials happening there. This is done over 100 trials, an average there showing that it does really excite the neurons that are there, and they produce action potentials. And that if you do this a few times in a row, so these are 5 pulses given in a row over 200 milliseconds, what you see is that they reliably activate, but the level of activity is kind of dropping. Okay, so it seems like it accommodates a little. Go ahead. And this is just a summary of these data, how big these are compared to the first. Go ahead, Stephen.
That optic fiber to what the confirmation that.
Oh, so every time we implant anything, well, we mean they, I don't think it's not talking about the potential did something. after the experiments are finished, you sacrifice the animal, you slice it, and you have to verify that the optic fiber was right over that region. Now, one thing I've learned from Kamran is that he is insanely anal about data from his own lab. And I mean, this paper took forever to get out. I mean, we did so many controls and anything that is not properly controlled, we just gets this started. We do. Go ahead.
The thing is, the activity lasts high for very long apparently. Is that due after the
This.
stimuli?
Yeah, so that actually I'm going to show you when I show you the phytophotometry data a little more. And here we just to confirm that these connections are actually real synaptic connections that are coming there. Here, this is done now in the same type of animal with channel rhodopsin, but now you slice the brain, you put it in vitro, and you record from SNC neurons with patch electrode. And what you see is excitatory postsynaptic current. So this is holding the voltage at some low level. You see a negative current, that's an inward current, which means it depolarizes the cell. That's the average of that current. This is to a single pulse. And this is the same current in response to 5 pulses. And those of you who took courses with me, you notice the short-term depression of this synapse, which is average there and is kind of correlated with the drop, the accommodation of the firing rate that you saw when we did this in vivo.
And these are presumably dopaminergic cells firing?
Presumably. So, but in fact, yes and no. Give me 1 second, let me finish this and I'll come back to that. So this synapse, when you hold it at low voltage, is blocked by NPQX, which is a blocker of AMPA receptors. And if you depolarize the voltage in the presence of NBQX, you still get a current. That remaining current is blocked by APV, which is a blocker of NMDA receptors. So it is a glutamatergic. Okay, it's fast, it's glutamatergic, and it's gone. Now, what you said, there are 2 factors to consider. One is what I told you. This is published, and it's not something that we have to verify. Of the neurons in the SNC, 95% are dopaminergic. Of the neurons that were recorded there, right, that get input, about half of the ones that we found labeled positive for tyrosine hydroxylase and about half did. Now, tyrosine hydroxylase is just an antibody to an enzyme that is in neurons that produce dopamine and norepinephrine, and we just labeled them. The antibody is as good as an antibody is, so take that for what it is.
Because you're saying half only?
But let me give you more evidence about the dopamine. So we are specifically interested in this case about the action on the dopaminergic, or at least in this case. But it was about the half of the recorded neurons that were labeled. So one of the things that we went after is of these cerebellar nuclei, there are 3 different cerebellar nuclei. And we wanted to ask, on each side, there's 3 of them, right? There's the dentate, the fastigial, and the interposed nucleus. Wanted to see which ones of these guys are projecting. So in this case, in fact, we're not distinguishing. So in this case, we infected these nuclei with this AAV1 virus. So this is adeno-associated virus type 1. And it's a Cree virus, and this is in a mouse that is a reporter of GFP. So that means that it's called an RCE or RCE LOXP mouse. In that mouse, all neurons have potentially the ability to shine with green fluorescent protein, but they will do so only if they have Cree in them. So that's why we're injecting Cree there, right? Now, the property of this virus is that it not only infects the neurons that you actually put target, but it infects the neurons that are once postsynaptic to that. So it jumps one synapse and integrate in the forward direction. This is simply showing that the fastigial, interposed, and dentate are all infected. This is what we infect, right? This is across several animals. This is the vestibular nucleus. So ignore that, not relevant too much to us. Now we go to the SNC. So this is the target neurons. So what's labeled here is green fluorescent what's shown here is green fluorescent protein, which is what we are using to figure out who is being infected postsynaptically. So you can kind of see the neurons that are green. In red is tyrosine hydroxylase, which is labeling, as I said, the dopaminergic neuron. So if you look in this case, this neuron shows both GFP and tyrosine hydroxylase, and you merge it and it's there. In this case, this neuron shows GFP, but no tyrosine hydroxylase. So it seems like it's infecting both dopaminergic and the non-dopaminergic neurons. The non-dopaminergic neurons in SNC are almost exclusively local GABAergic interneurons. So that's potentially those guys. So that's basically showing that what we find is, yeah, actually, I'm not absolutely, oh no, this is, I'm sorry, these are slices of different parts of the substantia nigra showing that neurons are labeled in all parts of the substantia nigra when we do this infection.
But what you're saying is then that from the cerebellum targets about half of the dopaminergic cells, and none of the others. In that way.
No, in fact, it probably is targeting a lot fewer than half. I'm saying of the neurons that are targeted, which are potentially a lot fewer than half of the neurons there, half are dopaminergic, half are not. That's what I was saying. Okay. So you can, to ensure that this is really one synapse away, you do the opposite too. So here we infect the SNC neurons with a virus that goes retrograde. So this virus infects the cell and jumps back one synapse and labels the neurons that are presynaptic to this. Same type of GFP reporter mice. And when you do that, this is showing that the infection actually, these are called starter cells. These are the neurons that we infected. And these are going back now in the deep cerebellar nuclei, showing that in all three of these nuclei, there's a whole group of neurons that are potentially projecting to the neurons in essence. This is not my strong suit. It took me a long time to actually learn all this language. There's a lot of these games you can play with these viruses. Go ahead.
It's too late now, I know, but do they not have a dopamine reporter line now? So they
In fact, all the rest of the stuff that I'm doing
because I'm like,
is
the
with,
cage vein is, I mean, it's not specific for dopamine and it's not gonna hit all the cells. So you had one with indoctrination.
correct, yeah. So that's the new stuff that's not in this paper. But all the other stuff that I'm going to show you, this is just a summary of what I'm showing you here, showing that in different slices of the deep cerebellar nucleus, you get plenty of neurons that are projecting.
So,
Go ahead.
the retrograde travel of that virus to the stoma, it confirms that it goes to cerebellum.
Yeah. Or that the projections are coming from the cerebellum there. So for the next part, I want to actually describe this method that we had just started, I mean, Conrad's lab had just started using this, and I got really involved in this, and that's called fiber photometry. This is the single color version. So I apologize. This is what I showed you before. So here you infected with channel rhodopsin and you were activating with a laser, but you can also, in this case, the example I want to show you is that you can infect anything with a reporter that shows you either activity or something else and see if it's active. In this case, in these same neurons that we had channel rhodopsin, this different group of mice, but in the same types of experiments that we had channel rhodopsin expressed in the cerebellar nuclei and had our laser there, we infected the dorsolateral striatum, which are the potential targets of these dopaminergic neurons, with a virus that expresses a reporter for dopamine called D-Light. Okay, so D-Light has a variety of versions. It's a modified version of the D1 dopamine receptor. So it gets inserted in the membrane, it binds dopamine, and instead of doing its actions on cyclic AMP and then on cyclase, it just shines green. Okay, so what I'm going to show you is a recording from these, and this I'm going to come back to. These are average data across different animals, this is showing that when you stimulate these projections to the SNC with a single pulse, what you get is a dopamine release at that level on average in the dorsolateral striatum, right? That fluctuation of dopamine due to that is very similar in amplitude to the spontaneous fluctuations that you get in the striatum of dopamine. But if you do a train of these pulses, then you can really get a lot of dopamine, okay? So in fact, it goes up and it goes below baseline after two. And we can do this also not just by one reporter. So in this case, oh, no, I'm sorry. Let me back up. I'm getting ahead of myself. Remember, channel rhodopsin, we're shining blue light and recording dopamine here in the neurons, the dopaminergic neurons here. So these are dopamine genetic mice, they're called DAT3 mice. These are mice in which the dopamine transporter is labeled with, you can label only the dopamine, the neurons that have the dopamine transporter with the virus that you're expressing. And we express a virus known as JAWS, a virus that's carried the construct known as JAWS, which is an inhibitory construct that is activated by red light. So if you activate by continuous red light and within that do the pulses of blue light, what you get is that the level of dopamine that you were getting is reduced by almost 60%. Okay, so this has already gone out, but we're repeating these experiments, but instead of using an optogenetic construct, we're using a chemogenetic construct to actually turn off all the dopaminergic neurons at once to see if we can do better than that. But what this is saying effectively is that the release of dopamine that you saw there really was due to the dopaminergic neurons that were there, right? That potentially are under or a lot of it was. Okay. Here's another component of this that you can do. Again, correlative component with R manipulation. Same type of animal, D-light there, channel rhodopsin there. We put them on what's called a circular treadmill, it's really, a cylinder. And the animals are head fixed, we can record or stimulate, and they run on this treadmill. And at the same time, you can record the dopamine release in the striatum. These are the green lines that you see there. And the animal's movement. This is a time scale of many minutes, right? The animal's movement is shown as speed. And in these sections that are shown in blue, we're shining blue light to activate channel rhodopsin, either as single pulses or 1 second trains with 9 seconds in between, or 3 second trains with 7 seconds in between. So there is a baseline fluctuation, you can see that, of dopamine. These are single pulses, and this is the average of these data. So it's kind of a running average that you see there. And here is just a zoom into these guys. So this is a 1 second period. This is a single pulse, you see that. 1 second train, you see this. 3 second train, you see that. This is in a single animal, in a single trial, okay? You average it down and you see what I showed you before. Single, 1 second and 3 second dopamine. And then you can actually ask if there's a correlation between the animals walking and the levels of dopamine that you release, and you get a pretty strong correlation between the two, okay? So far, everything I showed you are stuff that we are doing. Now, the question is this system active during the normal behavior of the animal, right? Without you stimulating it and whatnot. So to do this, we do really simple behavioral tasks to test this. One of them I just showed you. First, all of this is in head fixed animals because we need to record, and movement artifact is, again, it's not a huge problem, but to remove movement artifact, what you need to do is you need to actually do your recording with 2 flashes of light, and it's just the resolution went down. So we did all this here with head fixed animals. Again, we're doing experiments now with animals in an arena. We did the locomotion as I showed you. We did a simple Pavlovian task in which you give the animal a cue, usually a light or a sound, and then the animal gets a reward, a drop of water, and it licks it, right? And the animal learns to do this over several days. Or we did a simple conditioning operant task, right? And here the animal gets it, and the animal has to move a lever. When it moves the lever, it gets a cue, and then the reward. So the cue is irrelevant here, it's the lever movement that leads to the reward. So here, what I showed you is us stimulating it. This is the free-running animal. So what we're recording in this case is we're recording the projections. Maybe I should, before I show you the data, explain this. Here, we didn't do channel rhodopsin. We expressed in the neurons of the cerebellar nuclei GCAMP7. GCAMP is calcium indicator, right? And it's, when it binds calcium, it fluoresces. So you can actually record that, and calcium that you're looking at is used as a proxy for action potentials here. So it's a proxy for activity. Here in the SNC neurons, this is a DAT3 animal. We expressed a different calcium indicator known as R-GECO, which is red. We have a single fiber above this, but we go back and forth. So this is dual color fiber photometry. So you can actually back and forth use, green laser, blue laser to activate these and record them one-on-one, and then demultiplex them to get them out. And what you do is you get recordings. So this is activity of the cerebellar inputs to the SNC, showing that as the animal moves, this is the animal's movement, the activity is going up and down. This is the activity of the dopaminergic neurons in the SNC, right? At the same time going up and down. And these are the average data. This is the onset of movement and the average data. This is many seconds, right? So there's nothing fast about this. These are slow recordings with photophotometry, volume recordings of what is happening. But clearly the onset of these is before that. Okay, that's the only point, is that potentially this is what is leading to that. Right. This is one animal. This is one trial, not this. This is average, I think, of. Good question. I don't know if this is average in a single animal across trials. I think that's what it should say. Not across animals. But we do that.
Just look at the timing. Is the idea that the substantial magnitudes are finding first, and there's a feedback loop?
No, in fact, they're going right on top of you. We don't have the resolution to show that. That's what we're showing here.
Yeah.
They're going right on top of each other, right?
You're not gonna have good control.
No, but in fact, we have additional evidence, and I can draw that about what, I mean, yeah, I mean, it looks like potentially even this is moving up earlier than that,
And
right?
that could be a feedback.
Yeah, you don't know, because this is in a whole animal. You have no idea what's going on.
So far the hypothesis is that this connection between cerebellum and brain and the potential anger control the speed of movement.
So far, our hypothesis was that it controls what, I mean, this has caused so much trouble using this word, but I'm gonna say it, the vigor.
Oh, yeah.
So when we say the word vigor, everybody has an idea about what this means, including me. And I mean, we had a lot of talks and discussions among ourselves about what vigor is, but the simplest way to define vigor in this case, I mean, vigor can be speed, can be acceleration, can be anything. But if a movement is happening, 1 over the time that the movement happens is called vigor. It's just 1 over the time that it takes for any movement to happen. Okay, you can call it speed.
It
Yeah.
plays speed with like sort of sprint, right?
Hmm?
It's a bit easy for speed. I would have thought it's a force measurement.
Exactly.
No, it's, I mean, Jasper has a whole book on this and the role of the cerebellum in vigor. And that's, I think, the first sentence that you define.
Writing a book doesn't make it work.
No, I know, but we are saying that it makes movements happen fast. Okay? Good. Okay. And in fact, huh?
Speed isn't
quantifying with speed, but you can quantify acceleration or what is the derivative of acceleration. Yeah. And that, in fact, is one of the major deficits in humans that is found in Parkinson's. Not that they can't do things, but it's like you slowed them down, right? And there's a really beautiful paper on that, looking at Parkinsonian patients and measuring what is wrong, having them play a computer game compared to controls. And that basically what it comes down to, how fast can they do this stuff, not that whether they can do it or not. Let's come back to the discussion points. I just want to get, not much is left of the data, we're almost there. So let's see. Oh, this is simply showing that obviously the activity that you see, the level of activity in these projections is bilateral, right? But of course the animal is running, so that's a bilateral movement. But to see what happens if you look at it in a unilateral movement, we could actually see that in our operant test. So I'm going to show you first simply the animal without any reward. It's getting habituated to the area. It's just sitting there and moving this lever while we're recording the activity of these projections there. And what you see is that as it moves the lever, this is back to front, so just movement of the lever that we average, both the left side and the right side activities go up, okay? The relative amplitudes are meaningless. In fact, I think the left side, yeah, so this is the right hand, so the right side should correspond to the left hand, but these are optical signals, so the amplitude is different. They are forced because of the lever that we're using to use that. And in fact, one of the critiques we got was what happens to the other hand, right? So it's not they can move it, right? So we had to go in, and so that's when Deep Lab Cut comes into play. So you can use the software Deep Lab Cut and go through all the movies and actually monitor what that does, and then take the sections that hand is clearly not moving and only analyze those. So we had to provide those data too, to show that even when it's not moving, that this is bilateral. Now this is interesting. The animal moves this, and then after a couple of seconds, it gets a cue and then a reward. Now, this is in the same animal. This is the signal for movement. This is the signal when the animal gets the reward, right? Which is huge compared with that. So that the animals are water. Yeah, the animals are water deprived. They get this and they have a huge response to just getting water. So the question for us was like, that's where we started playing with photogrammetry is, what is this? So is this really the cerebellum sending a reward signal to the substantia nigra? Because reward processing, go ahead.
So do they sometimes get the reward or sometimes get the cue? Because if you think about clicker training, the cue should have given the video, the dopamine, not the water.
Yeah, so the cue is here. And 1 second later is the drop of water and reward. But this is not dopamine. This is the input from the cerebellum, right?
But some of it,
So, in fact, no, it targets dopaminergic cells. But this input is what the cerebellum is telling these cells, right? In fact, I'm going to show you, we recorded them simultaneously, but I don't think it works.
So that's got that. So both of these are deterministic. So if they push the lever, they get a cue, and they always get cue, always.
In this experiment, yes. So the question is this really still movement? Because even when they get the reward, they have to consume it, they have to use the tongue, and that goes back and forth. And could it just be the movement of the tongue that does this? So,
Oh my God.
So we switched to the simplest task, the Pavlovian task. And in fact, we spent a year and a half doing this in a way that I'm not too happy, but let me just show you. We wait for the animal not to lick the spout which is in front of it, because the animals are just curious, they lick. But they have to get at least a 3-second interval of no lick so we can get a baseline. If they do, then they get a cue, right? And they get a reward, but they're rewarded in 90% of the trials, and we skip the reward in 10% of the trials. This is an example of a raw recording while the animal is licking the spout. So this, it's not very fast, I apologize, but each of these dots is when the animal's tongue is touching the. Yeah, I apologize for, oh, is that better or no? It's good on my screen, it's the Zoom part that's slow. It's good enough, right? You get the idea. So you get the fluctuations and the animal tongue movement, which shows that.
But what is it?
There's no-
showing?
There's no-
Licking. That's the licking activity.
This is the recording of the cerebellar inputs. The licking in this case is only detected by the animal's tongue touching. This is not with deep lab cut here. So now everything we do, I mean, we still have the licks detected there because you have to detect that the animal is no licking in real time, right? So that is with the contact and vibration, right? But now we detect the actual behavior licks with deep lab cut, which is.
So the point is not a motor signal, it's a word signal, because you don't.
I haven't shown you that evidence yet. The question is that right now.
It's not peaking when they click. It's only peaking when the lips have a word. Is that what the video is?
No, so here's an example. So here's in one animal across 150 trials, the licks. So here is the cue and the reward at the same time. When the animal gets the cue independent of reward, it starts to lick, right?
Yeah.
If there's reward, it licks for longer. You still get a signal with licking. You just get a bigger signal with reward. So here we did the next best thing. So it's still not clear. You can't disambiguate this, whether it's all just movement. So what we did is we switched to this same task because we had the animals. We put them in a task where we gave them a regular reward, drop of water, or drop of sweet water, which we call high reward, same volume, or we omitted each 1/3 of the time. And what you see here, this is simply licking. This is one animal, this is across 8, okay? Is that the animal starts licking. If there's no reward, it stops after a while. But if there's a drop of water, the licking continues, whether it's sweet or not. And in fact, on average, the licking is identical for the first 3 seconds, but starts diverting. If it's sweet water, it continues licking a little bit, okay? But when you look at the actual signal coming from the cerebellum, this is again one animal that's average across 8. There's significant difference between these from second to 1. And here in these animals, we also recorded the activity of the dopaminergic neurons, both in the soma and in the striatum. I'll show it to you, you make of it what you want. So this is the dopaminergic neurons, in fact, clearly respond to reward as they should, they're supposed to, right? However, it looks like, I mean, first of all, the axons are different than the soma, potentially because what we're recording here is just whatever fraction of the soma we're projecting to that location. It's a somewhat different signal, but I don't know if I want to say anything more. In fact, we didn't say this in this paper, but if you measure this, put a line at 0, the dopamine signal in the unrewarded animal has a negative component at that point that is significant. But we have better experiments right now. So we have new experiments, which I'm not going to show you today, that we separate the time of the cue from the time of the reward by 1 second, which we should have done from the beginning. But the animals were just not learning even in 6 weeks when you separated these two by 1 second, right? It was something that a rat learns like in a week. These guys were not learning it, but eventually they learn. You know, you just have to persist. Another analysis that we did is in the paper, but I'm not showing it here, is going in individual traces and looking at the correlation between the size of the signal and that, and see if the slope of these correlations is changing, and it is significantly different. And despite the fact, yeah, that's a fine. Go ahead. Between the size of the signal and the level of licking in individual trials across all animals, right? To show that this significance that I'm showing here, despite no change, there is really a difference that is due to reward. Even then, one of the reviewers said, well, how do you know it's authentic? Right? I mean, one is a drop of plain water, the other one is a drop of sweet water. It could be just sweet. That makes that signal good. So what we did, again, not published, but I'll show you the data next time, is that we did a devaluation. So we compared the animal signal when the animal was doing this in water deprived, or after giving it a milliliter of water, same animal, right, in a different day. And there's huge difference in that signal despite identical licking when it actually licks and gets the reward. So you can devalue the signal and get that independent of how much it's licking or whether it's water or sweeter. So in conclusion, I basically said that the SNC dopaminergic neurons get a direct connection from the cerebellum. The input is bilateral, even when movements are unilateral. It potentially has both movement and reward value information. There is increased release of dopamine due to these signals, and it is contributing to quote unquote vigor. Okay. Now, what does this mean? So you may have heard that when people have Parkinson's, that one of the issues is a loss of dopamine, as I told you, in the striatum, right? Dopamine replacement therapy has been known for decades to work and give the patients L-dopa, which is the precursor to dopamine. And that works for a long time until it doesn't work, right? One of the questions I ask my class, my addiction class, is why don't you just give them dopamine? Why do you have to give them the precursor? Like the blood-brain barrier. Okay, so it's worked, but surprisingly, until relatively recently, nobody has directly shown that it is the dopamine release from the substantia nigra directly into the striatum that is required for these movements. That's a relatively new thing. It's very surprising. We think that that would be known, but it wasn't until the event of optogenetics and all of that stuff that people started to show that dopamine is really important for the proper initiation of movements. And I have the papers up there. So it is also known that inhibiting the cerebellar nuclei slows reaction times in motor tasks. So that has also been known. And activating it actually produces movement. So you can activate the cerebellar nuclei by inhibiting Purkinje cells, which are projecting to them. Purkinje cells are GABAergic, so they're always inhibiting the cerebellar nuclei neurons. So if you activate them, they just push them down, and that initiates movements. Now, our idea is that the cerebellum, in a way, as I said, it's like a second brain. It has this really unique property among all structures in the brain that it gets information from all sensory inputs and all over the cortex. It is informed about everything that is happening in the body. So potentially, if there is movement, it can send a signal to go. If there is a reward and it needs to make that movement faster, it sends a signal to the SNC that this is the time to go. That's our idea, because effectively, I've come to think of the cerebellum as a prediction machine. It's just predicting everything and then matching it with what you're trying, with the brain, the rest of the brain is trying to do, and trying to optimize. Now, be it sensory or be it motor. That's what it does in fish, and that's what it does in people. So one of the things that we put in a proposal that we sent, which was scored but not funded, was we wanted to check in a mouse model of Parkinson's. So there are these mouse models which are, I mean, there's a lot of ways you can produce Parkinson's in mice. One of them is to just, inject the midbrain with, 6-hydroxydopamine, 6-OHDA, right? And that basically goes in and kills all the dopamine neurons, right? And the mice become Parkinsonian. But that has not been, that is kind of not used as a real model of Parkinson's. Now they use this model that goes after the mitochondria and the dopamine neurons and disables the ones specifically for the SNC, because the reason that the dopamine neurons seem to die in the SNC but not in the TA in Parkinson's patients seems to be the overabundance of these mitochondria for some reason in dopaminergic neurons of SNC. And one of the things that one can do in people is you can do transmagnetic stimulation of the brain, right? Just basically put plates and magnetically stimulate the brain. And the cerebellum is one of the easiest places to stimulate, it's sitting right here, right? You're gonna have a hard time stimulating anywhere near where the dopaminergic neurons are, because they're sitting really in the center at the very base of the brain, right? So in our experiments, what we wanted to do was simply to see if you get these mice and you just stimulate this pathway, will you recover some of the function, which is a potential thing.
Oh, why wouldn't you? If the dopaminergic cells are dead.
The dopaminergic cells don't die like this. So they
Right.
start, first the terminals start to deteriorate in the striatum, and then eventually the cell body. But even their projections to the right nucleus node right next to them, SNR, even if you activate those, you can recover some function in the animal, even not the striatum. Nobody really knows. I mean, these are questions you would think people know. I mean, how the hell does L-DOPA help if the terminals are gone? Nobody knows. It's not like these are simple questions that people know. Nobody
But
knows.
moreover, I would like to do that. The L-DOPA treatment for Parkinsonian people is just floods the brain with dopamine. There's no timing cue.
No, there's nothing. No.
So what's the timing, what's the relevance of timing in your results there?
So that's not a Parkinson's question. That really is a science question. So the question is, what is the relevance of timing in the fluctuations of dopamine that we see in the striatum? And in fact, as there are experiments kind of like FARS where you can actually align things and show that things like bigger change. But the real answer is nothing. As far as anybody can tell, all that is needed is a baseline tonic dopamine level to recover almost all people.
You know that even stronger with deep brain stimulation.
Yeah.
So in deep brain stimulation, you use 1 hertz, and then you maybe, there is some timing effect because you do need to tweak it. But once it works, there's no information. The information is just click, 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 click. And that really supports very strongly your new approach, which would be shallow brain stimulation.
Yeah.
It would be very, very nice for the patients not to have to go through the skull and insert.
But that's only
Going deep in the brain, go in the back of the head.
But that's only good at the beginning of the disease.
Potential. Everything.
In Parkinsonian patients, can we bypass dopamine and directly stimulate what was that dorsal striatum and
No, so dopamine, not easily. So in fact, in deep brain stimulation, they go after the what is the excitatory nucleus in the
What are those global cells?
no, not the thalamus.
Because there's a lot of different sites for DBS.
but it needs cerebellum.
So here's the simple answer. The striatum has, again, almost 90% of the neurons are medium spiny neurons that project out. Either go to the pallidum or go to the substantia nigra pars reticulata. So in humans, it's mostly the globus pallidus, that's the output. In mice, it's mostly substantia nigra. But there are 2 groups of these neurons, right? They fall into 2 categories, and they're identified just by what? Whether they have D1 receptors or D2 receptors, right? Because the D1 receptors are the direct pathway, which is the go pathway, the D reaction, the D2 ones, or the no go. And he wanted me to do exactly opposite things. One is increasing sensitivity, the other. So it's not clear you can go in there and simulate shit and may get all sorts of weird stuff going there. Right? So,
What normally L stops working to, in terms of those dopaminergic neurons? Are they just-
you can, there's nothing you can do other than different subthalamic nucleus is what they say.
Okay.
The subthalamic nucleus, deep brain stimulation seems to still work in that. Bypasses all of the dopamine that goes to stimulate an excitatory nucleus that's part of the immunity pathway. Again, no one knows why this stuff works.
Yeah.
And part of it is that all of this stuff that I'm telling you about the basal ganglia is really a very crude first order approximation of what it's doing, right? It's doing a lot more than that and doing a lot more complicated stuff. And we just wave our hands around. So all of this direct and indirect and no. The first order, yeah, that's what it does.
No, Cameron, right? His lab showed that previously projections from cerebellum go to VTA, that
Yeah.
was first paper. Now you show that cerebellum to
That's
substantia nigra.
Yeah.
So these are both VTA and substantia nigra are dopaminergic nuclei.
Yeah.
So where VTA projections dopaminergic?
So the VTA projects to the ventral side of the striatum, which is known as the nucleus accumbens. And that's the reward pathway. And there's also another pathway that goes to the prefrontal cortex. So those are known as the mesolimbic and the mesocortical pathways. And those are clearly involved in reward processing and addiction. An addictive drug is defined as a drug that, among other conditions, has to activate the mesolimbic pathway. Otherwise, it's not addiction.
Two separate learning.
Completely separate.
Yeah.
So, yeah, so I'm working with another postdoc who is recording nucleus accumbens. Again, has two parts. There's a core and a shell, and they're thought to do different things. But it's really cool the things that we can. I mean, one of the things that we showed yesterday in lab meeting, this postdoc, his name is Jorge. So is that he showed dopamine release simultaneously in the accumbens and the prefrontal cortex by activating the ventral tegmental area neuron. Accumbens lab is not dopamine / basal ganglia lab, but apparently nobody's done this. Actually, record dopamine release simultaneously. Completely different dynamics.
That's something we haven't done this experiments, right?
Yeah, I mean, you would think that, but I mean, I'm sure people are doing it. It's not just, he's like, I don't have these data, let me get it. And he showed it yesterday. Of course, Kamran said, I don't believe any of it, so.
I guess you already said this, but the L-DOPA treatment
Yeah.
experiments completely go against the idea that there are these two paths. One is activation in the striatum or activating and suppressing activation of movement.
But both of these are enhanced by dopamine, by the
Why do you go against that?
way. It's also
Yeah.
against.
Because, because it's spread everywhere and it's not.
I don't know where.
L-DOPA.
But they have
Dopa is taken up by the
Dopamine.
live dopaminergic neurons to make dopa.
Yeah, it has to be taken up by the dopamine neurons to be converted to dopamine and be released.
So.
The interesting part is that if the terminals are dead, who's taking that? Converting it to dopamine. It's like, that doesn't make sense, but
And
no,
it's
it's
for
still.
anyway, where they have different receptors that are gonna be different.
Anyway, thank you all for.
I will do that.